situation, we can think of many microorganisms which is more dangerous than virus. And here the biologists come into the limelight. Here comes the role of microbiologists to understand more about the organism. And today, the microbiology and microorganisms reached every nook and corner of the world. Even the common man speaking about these tiny organisms. The better understanding of this problem lead to the development of the better solution. Microorganism means it is not always disease-causing organism, but there are useful organisms. Without the microbe, no life in this planet can play a key role in the environment. In this context, Department of Studies of Microbiology, in association with Vignan Bhavan, and also in association of Microbiologists India, and the Mysore chapter is organizing one-day international webinar, Current Perspectives in Microbiology. I very happily inaugurate this international webinar. And among today, there are speakers, Ram Shivan, Associate Professor, who is from USA, and he is speaking about, he has worked in HCV, HIV, and Zika, and influenza viruses. And this group is extensively worked on the regulation of immune genes and also the impact of the viral infections and autoimmunity. So I have gone through his biodata yesterday, very extremely impressive. I congratulate him and also thank him for accepting to speak in this occasion. And there are other pioneers in the field, which is evident in the publications, which is peer-reviewed journals like Nature, Blood, etc. Today's topic, heterogeneity and human responses against viral infection determined by the host is by the sovereign. So there are three speakers. Another one is from Dr. Malaysia, Dr. Gokul Shankar Sebastian, and from MST University, Malaysia. And he's speaking about the food safety and related things. And also another one is Angel Medina, and he's from United Kingdom. Definitely, this department has worked well. 3 different countries, 3 different eminent scientists are speaking. Really, all our participants are very lucky to share the thoughts by these eminent peoples. On this occasion, reminds me a quote by the renowned scientist Louis Pasteur: "Science knows country because knowledge belongs to what we call humanity." Science belongs to science. Know that no country, science knows no country, but belongs because the knowledge belongs to the humanity and is the torch which illuminates the world. So I wish the deliberations, and I encourage this department to come up in many scientific programs. Let this webinar give a good deliberation. So let it be useful to the society. All the researchers should be useful to the society. Now today, because of pandemic, so we are thinking, so every research somewhere what we call useful. Since 8 months, we are what we call finding out some what we call vaccine for the pandemic. So that is microbiologists and other scientists definitely work on that. And any society always should reach the society. This department is doing. I wish you all the best. And I congratulate the chairman and all the professors of this department for conducting this. Thank you very much.
Thank you, sir, for your inspirational and supportive talk. Now, coming to the brief introduction of today's speaker, Professor Ram Savan. I'm very happy to introduce Dr. Ram Savan. He graduated from College of Fisheries, University of Agricultural Sciences, Bangalore. That's where we got to know each other. He received his PhD in 2004 from Kagoshima University in Japan. He completed postdoctoral training in the Laboratory of Experimental Immunology at the National Cancer Institute in 2011. He joined the Department of Immunology, University of Washington as an assistant professor in 2011 and was promoted to associate professor in 2017. He is bestowed with many honors and awards. To name a few: 2001, Monbusho Japanese Graduate Fellowship Award; 2004, Dr. V.G. Jingran Best Thesis Award; 2004, JSPS Postdoctoral Fellowship Award; 2005, Dr. T.V.R. Pillai and Dr. M.V. Gupta Best Overseas Scientist Award; 2008, Federal Technology Transfer Award, National Cancer Institute; 2009, Directors Intramural Innovation Award; NCI, 2010 Cancer and Inflammation Postdoctoral Project Award, NCI, 2010 Milestone Young Investigator Award, the International Society for Interferon and Cytokine Research. He is serving as the member in many professional organizations, American Association of Immunologists and International Cytokine and Interferon Society, RNA Society, and American Society for Virology. Ram Savan's team has been at the forefront of genetics and biology of interferons, mediating viral restriction, mRNA splicing-induced diversity of interferon-activated proteome, and control of immune responses through non-coding RNA. His pioneering findings have been published in highly reputed journals like Nature, Nature Communications, Nature Immunology, Immunity, Blood, etc. So we are happy to we are very happy that you are here with us, and we are very excited to listen to your talk. Over to you, Savan.
Thank you. Can you hear me?
Yes.
Okay. Thank you, Vice Chancellor. It's been an honor to give this talk, and Dr. Shubha Gopal for inviting me. It's a great honor to be here and talk to AMI. So I will share my screen. Hopefully this will work out.
$150 members here.
Very good. That's how we can reach the people through technology.
Sorry, give me a second.
Department students on the IFNL, of course.
Sir, we are not in the full. Yeah.
Okay, can you see my screen?
Sure.
Yes.
Okay. So today I'll be talking about the heterogeneity in our immune responses, because, you know, when everyone gets infected, some people clear infections very easily, but others don't. And my lab has been really interested in this question for a long time. So before I get into the talk, I just want to acknowledge all the people who have worked. As you know, I don't pipette anymore. So the people in my lab have been really important for all the work which I present today. So most of the work I'm presenting today is from Adriana Ferrero, Johanna Schwerk, and Frank Sovik. So, and without our collaborators from the Michael Gale lab, Michael Gale, Alison Kell, who have put a lot of effort in this work. And also I'll be presenting some data from Matthew Doherty's lab, from UC San Diego, Jenny Hyde, as well as other collaborators around who have given us some reagents. Of course, the funding from NIH and the German Foundation. So if you just consider the vertebrates in evolution, you see innate immunity kind of originated around 550 million years ago. And this innate immunity immune system is very primitive. Like you have macrophages and interferons. And this was the time where these started in these vertebrates. As the evolution progressed, once you saw bony fishes, you also saw interferons as well as adaptive immune systems like B cells, which make antibodies, and T cells, which are important for adaptive immune system. And I'm sure most of you have heard interferons because these are important mediators of antiviral immunity. Especially interferons have come into play in SARS-CoV, and I'll be talking a lot about interferons in this talk. Adaptive immune system, when you talk about adaptive immune system, you have B cells, and B cells make antibodies, and vaccines are based on B cells and antibodies, and those memory responses where when you see the virus again, you generate these antibodies, and these antibodies neutralize viruses. So what is intriguing, right from lamprey to humans, there are plenty of pathogens, including bacteria, virus, fungi, and parasites. And these have been there right from the beginning, right? And so what you see here is all these organisms have been fighting these viruses, right? Or bacteria or fungi or parasites. So this immune system has kept these pathogens at bay so that we have not extinguished ourselves, and we're constantly evolving. So if you think about humans, for the longest time, we have also been around pathogens. For example, smallpox, right? So when you have so many viruses and bacteria and other pathogens, we still are not extinct. That means to say our immune responses are really strong, and they're fighting these pathogens for a very long time. So my lab is kind of interested in understanding what does our DNA tell us about how we fight these pathogens. And just to give a primer, and I'm sure most of the students in the audience know what a DNA is, and DNA is made up of A, G, C, and T. And DNA gets transcribed into RNA, and RNA is the messenger molecule which gets translated into proteins. So if you step back and think about all these animals, right, from vertebrates to humans, what you see is most of our DNA are very similar. And if you compare, for example, line up these nucleotides and compare animal X, for example, here I'll say human, to animal Y, fish, what you see is there will be single nucleotide changes, right? So if you compare our entire genome, the human genome, to the fish genome, what is intriguing is 70% of our genome are very similar. That means to say, a lot of conserved genes, like the genes which are running a cell or running an organ or forming an organ, are all very similar. And if you compare humans to chimpanzees, 99% of our genome is similar. Just 1% difference between chimpanzees and humans. But now, if you compare human to human, 99.9% of the genome is similar. That means to say, the difference between the diversity among us is based on that 1.1%. That means to say our skin color, our hair color is all dependent on that 0.1%. And my lab is trying to understand how that 0.1% difference among us is important for warding off pathogens, or how we deal with pathogens. I might deal with a pathogen differently than you, and we are trying to figure out, is this encoded in this 0.1%? So to give you a much more deeper insight, for example, if you think about a gene, which is an mRNA, which encodes a protein, this difference, that one nucleotide difference, could be in the 5' UTR, or in the protein coding region, or in the 3' UTR. You don't have to worry about all these. But what you can see here is having a one nucleotide change could be very benign. There could be no difference if you had that one nucleotide change. Sometimes, if you have that one nucleotide change in the protein coding region, it could turn out beneficial, meaning making that protein more robust, making the protein fight a pathogen. Or sometimes you get a change which is bad. For example, maybe you will end up not making that protein, and that protein might be very important for the survival of that person, and that could be lethal. So what we are trying to figure out is, are there any changes like this in the human genome, especially in the immune genes? And if these changes, would they affect us how we fight pathogens? So my lab is interested in this fraction of 0.1% difference, because the 0.1% difference is in the entire genome, but we are just looking at a handful of coding immune genes which are important for immune responses. So before getting into the subject of innate immune responses, the first immune response, what you see, this is way before B and T cells come and protect us. This is called innate immune response. The minute the pathogen is there, maybe 15 to 20 minutes when the cell sees the pathogen, innate immune responses are induced. And one of the major innate immune response is through interferons. So the name interferons was given because they interfere, and they interfere with viruses. So these are the first and critical defenders which clear infections. So there are two major interferons which have antiviral activity. One is type 1 interferon, and the other is type 3 interferons. And you don't have to worry about the differences of these interferons, but what you need to know is they signal into the cell. This signal is telling the cell either your cell is infected or your cell is next to an infected cell, and you need to get ready. You need to increase your defenses so that you neutralize that virus. So what these interferons do is activate certain signaling pathways and induce hundreds of interferon stimulated genes or ISGs. And later in the talk, I'll be talking about certain ISGs, but what these ISGs do is go and block viruses. So it's a very simple system. The minute your body sees a virus, it activates this pathway so that you can block viruses replicating. But viruses are also very smart. They have figured out ways to neutralize this. So the question is, because we are still surviving with number of viruses in this world, that means to say there is a constant battle between interferons or innate immune responses and pathogens, and this is called as host pathogen race. Okay, so when you get infected, for example, you had influenza, right? And you feel bad, you feel fever, you have chills, you have headaches, and that is because because of interferons. It is not the viruses which give you fever, chills, and headache. It's the interferon which gives you fever, chill, and headache. So, but the good news is interferons have these ISGs. They're your foot soldiers fighting these viruses. So I'll give you one example how one nucleotide change in our genome can differentiate between a person getting infected with hepatitis C virus and clearing it without any treatment, and other people who get the virus and who cannot clear, who end up with cirrhosis and cancer. So hepatitis C virus, as you all know, it's a common bloodborne infection. It is prevalent throughout the world. And only in the US, there are 3.2 million infected people. And worldwide, there are 170 million people who are infected. And the current estimates, and this pales from what is happening with SARS-CoV-2, but still there are 8,000 to 10,000 deaths per year only in the US. And there are new drugs, but the drugs cost $120,000 per person. So this is a problem. And if left untreated, and hepatitis C virus is not like the acute influenza virus or acute SARS-CoV-2 virus. Hepatitis C virus is a silent killer. It takes 30 years from when you are infected to having an end-stage liver failure. So what happens is if you leave it untreated, it goes from chronic hepatitis to having a cirrhotic liver, and if left untreated, you end up with liver cirrhosis, with liver cancer. So, but one thing was really exciting in the field was there were certain people who would clear the virus or who would be treated with interferons and they would clear the virus. But there were a subset of patients who just could not clear this virus, even with interferon treatment. And I told you, interferon treatment is a hard treatment because you end up feeling sick for a long time and still they could not clear the virus. So so a number of groups were trying to figure out what is causing this difference between these two populations who can naturally clear versus who cannot clear. And they did something called genome-wide association study. So genome-wide association studies is basically sequencing the entire genome, all the polymorphisms in the genome, and looking for any signal which will differentiate from a person who cleared the virus versus a person who doesn't clear the virus. And you can see this is all the genomes lined up, and it's usually called the Manhattan plot, because if you've gone to New York and you've seen the New York skyline, it's the Manhattan skyline. And usually what you see is a bunch of signal, and which are the signals of the genomic, the bunch of genomic signals what you see. But here, you see that there was just one strong signal in chromosome 19. And this one nucleotide, just one nucleotide, determined people who cleared the virus versus who did not. And for example, in this case, people who cleared the virus had a C in the chromosome 19, and people who got sick, who could not clear the virus, had a T. So my lab and other labs worked for a very long time. And this was one of the first projects we started as a lab. And what we found was there was a polymorphism in a gene called interferon lambda. So this is the same type 3 interferons. And this SNP induced more interferon lambda if you had a C. And what happened there is if you have a C, you cleared the virus. But unfortunately, if you had a G or a T, what happened is this interferon lambda was not made and the virus proliferated. So that one nucleotide had an enormous effect on how you clear the virus. And this was kind of a very important observation, because now you can think the people who want to go and get the interferon treatment or other treatments could find out what are the odds of clearing this virus. And one of the ways they saw that was looking at the frequency of the good genotype, or the C frequency. And what they saw is the C frequency was the highest in East Asian Americans and European Americans, meaning they cleared the virus really well. But if in African Americans, the C frequency was very low, that means to say they could not clear the virus. But there were still some African Americans who had the C, who could go on these therapies. So you can see how just knowing the changes in the genome can have an enormous effect in administering drugs in these cases. So, so there were a couple of other SNPs, which my lab and other labs around the world have figured out. And what we basically see is G is really important for clearing hepatitis C virus. So this is one example, but going back into a bigger picture, because we are talking about interferons and we are talking about pathogens, we just wanted to know, I said that we had two interferon systems, and why do we need two of them? And these two interferon systems are there right from fish. That means to say we have not lost it. That means to say it has some really important role to play in protecting us from viruses. So the type 1 interferons are critical for all viral infections. But what we see is type 3 interferons are only important for certain viral infections like hepatitis C virus, noroviruses, and influenza virus. And we have been very interested in understanding if they have any specialized functions, and because the biology of these two interferons have been poorly understood. So one of my postdocs, now she has her own lab at Ohio State University. She's a great PI. If someone wants to go there, you should contact her. She's a wonderful mentor. So she wanted to figure out what do these two interferons do? So big picture, both interferons are important, and in certain cases, they're collaborative, meaning type 1 and type 3 interferons both act on these viruses to kill this virus. For example, I'll give you a very quick example, VSV. VSV is an experimental virus. We use this to ask some basic genetic biological questions in the lab. And she pre-treated cells with interferon, which is type 1 interferon or interferon beta, or type 3 interferon, which is interferon lambda, and then asked if the cells were protected from this viral infection, because this virus can kill the cells. What she found is interferon, type 1 interferon had an early effect, but that the protection effect waned off post 48 hours. Type 3 interferon, in the other case, had low effect early on, but had a significant effect later on. And as you can imagine, if you add both the interferons, you are protected for a longer time. So this shows a collaborative effect. But what is more intriguing about type 3 interferon compared to type 1 interferon is type 1 interferon brings in a lot of immune cells to the site of infection. For example, in mouse, when we gave either type 1 interferon or type 3 interferon to the lung, what we saw is type 1 interferon or interferon beta brought a lot of immune cells there. And usually immune cells are good to clear up the virus, but if you have high infection, and if your immune cells are going to the lung, it is not good. It causes inflammation. And that's what is going on in SARS-CoV-2, because a lot of people are having high inflammation, and uncontrolled inflammation is never good. But what you see in type 3 interferon, that those immune cells are not coming to the lungs. That means to say type 3 interferons can fight the virus without causing inflammation, versus type 1 interferons who bring in these other cells, which are also important to fight the infections and get your adaptive immune responses going on, but they also do cause inflammation. So this story was published recently. If you're interested, you can go and look at Ferrero's paper. But what was really cool is a very famous artist from Bombay came up with, we were asked to put a cover for this paper at Immunity, and a famous artist in Bombay came up with this nice depiction of this, where she calls it Sagar Manthan, where the red is inflammation from type 1 interferon, and the blue is there's no inflammation, but it is type 3 interferons, and both kind of work together in collaboration and clear the virus. So how is this important in SARS-CoV-2? As you know, SARS-CoV-2 causes COVID-19, and there has been some data saying that SARS-CoV-2 is good at blocking interferon, but there is also some data that type 1 and type 3 interferons are also made. Now they're trying to use this in the clinic. There are certain data which just came out saying that interferons could be bad for COVID-19, but I think this jury is still out. There are some clinical trials going on, which will be more definite. But I do think early intervention with these interferons might help or may be key in controlling this virus. Okay, so I talked about interferons in a general term, right? But what do interferons do? Interferons induces multiple hundreds of immune genes, and these genes are the foot soldiers controlling these viruses. How do they do this? So I'll give you two examples, and these examples are also important in the context of changes in the DNA and alternative splicing, and how these alternative splicing are important for controlling viral infections. So just stepping back, certain viruses, the viruses which we all care about, mostly they are RNA viruses which we care about. There are certain DNA viruses which are tumorigenic, which are also important. But in this case, the RNA viruses like flaviviruses, like West Nile, Zika, and Hepatitis C virus. They get into the cell, and they're very smart viruses. What they do is once they get into the cell, they try to hide in the cell. So what they do is they borrow membranes from the endoplasmic reticulum or the Golgi, and make nests for themselves, like a house, right? And they hide their viral particles in there so that the cell does not see them coming in, or the immune system of the cell does not recognize them as foreign. So they wrap them on, themselves around using these intracellular membranes and hide in these what I call as hives. Okay. The other example is alphaviruses, chikungunya, Sinbis, and I'll give you one good example for alphaviruses in this talk, as well as one example for flaviviruses. So what alphaviruses do is go and hide into the membranes of the lysosome, right? And you can see that the viruses are hiding in this, and they replicate in this region. And picornaviruses like EMCV, Coxsackie, or polio, they use the Golgi membrane to hide themselves. So now the question we have is, when these viruses hide in these organelles, how do interferon-induced genes find them? Okay, so before getting into the nitty-gritty of the stories, I just want to give you a primer on alternative splicing, because it kind of becomes important later on. Alternative splicing, as you know, in human genes are usually divided into exons, and certain exons come together to make the mRNA and the protein, but there are different combinations of this. Some, for example, splice form A uses the blue exon, the brown exon, and the green exon, but splice form C uses all the exons, and splice form B uses just 3 other exons on the right? But do these have effects? Yes, they have biological effects. Some have modulate the dosage of the activity. Some have new functions, which I'll talk today, and some lose their function. Okay, so the first example I'm giving is ZAP. It's called zinc finger antiviral protein. And then, as the name suggests, it is an antiviral protein. So meaning it goes and fights against a virus. So the zinc fingers are important to find the viral RNA and catch it, and then degrade the viral RNA. If you degrade the viral RNA, your virus cannot replicate. So this has been known for the past 20 years. There are multiple labs across US and in Japan who have been working on this. And what they found is ZAP binds to the viral RNA and degrades it. And what was also known is these ZAP is particularly really important for alphaviruses. Like I said, Sinbis, chikungunya was really important. So my lab, and I won't go into details about the entire story because it is recently published, what we found was there were 2 different isoforms of ZAP. There was a ZAP short because it's shorter in length, and ZAP-Long, which is longer because it has an extra PARP domain, which is inactive, but also a very curious 4 amino acid at the C-terminal called CAX-box. Okay, what does CAX-box do? CAX-box gets phosphorylated, and then a lipid moiety is added to it. That means to say it becomes hydrophobic and goes and gets stuck in the membrane. So we asked, between these 2 ZAP isoforms, is there a localization difference? Because this looks like it goes into the membrane. ZAP short does not have that CAX-box. And what we found is ZAP short is in the cell all over. You see the red, it's all over in the cell. It's not in the nucleus, but all over in the cell. But if you see the ZAP-Long, you see that they are in the punctate regions, right? And if you superimpose both of them, they are in 2 different locations, which was intriguing. And we did some super resolution microscopy to see that ZAP-Long is kind of coating on some of these vesicles, and ZAP short is all over the place. This was very intriguing to us. So long story short, we found that ZAP-Long specifically coats the endolysosomal membrane. And this was figured out because we did staining for LAMP1, which is important and which stains the endosomal membrane. But now if you see ZAP short, it is all over the place, but you see it is not in the endosomal membrane. But now you go and mutate this CAX motif, just the cysteine, and now see what happens to the ZAP-Long. It acts like ZAP-short. It is all in the cytosol. But if you add the CAX motif to ZAP-short, it slowly starts getting into the endosomal membrane. So why am I telling you this? Because if you look at alphavirus in our cell, within our cell, what you see is alphavirus are lined up in this endosomal membrane, right? And they're actually spewing out their RNA, and that's how they replicate in the cell. And when they're inside here, the innate immune response cannot see them, right? So, but our innate immune system is really smart. And what it does, like I showed you, ZAP-Long is coating this region, waiting for the RNA to be spewed out so that it can catch and degrade the RNA. So is this right? And we could show that ZAP-Long, and I'm not sure if you can see the double-stranded RNA. Usually when you stain for double-stranded RNA, it's the viral RNA, because host RNA don't have such double-strandedness, but they are kind of exactly on top of each other. That means to say ZAP-Long is in the double-stranded RNA where the replication is occurring, and they're chewing up the RNA. And ZAP-short is all over the place, and they cannot kind of go into those regions where the viruses are actually replicating. So is this important? Yes, because if you express ZAP-Long, what you see is you can really control the virus, right? ZAP-short also does control the virus, but it's not as good as ZAP-Long. But if you go and mutate the CAX motif, where now you make this cytosolic look like ZAP-short, now you can stop controlling the virus. But if you add the CAX motif, you start controlling it. So just 4 nucleotides at the end of a protein takes this protein to the endolysosomal membrane, waiting for the virus to infect the cell, right? And we have shown it in different ways, but again, you see ZAP-short does not have an effect, but if you knock down ZAP-Long, the viruses start replicating. So what I want to say is ZAP-Long acts predominantly as a viral restriction factor for alphaviruses, and it does that by alternative splicing, and the ZAP-Long is always being made in the cell, right? And it is always sequestered into these endosomes and waiting for the virus to get in. And once the virus gets in, it chops up the virus. So this is a good example of, there are hundreds of these ISGs. This is one example of how this one ISG specifically restricts alphaviruses. Is this ZAP important for another virus like EMCV? No, because EMCV does not use this endolysosome system to replicate, right? So I'll give you another example in the last few minutes, is how there's another ISG which is important controlling viruses like West Nile, Zika, EMCV, and we think this is also important for SARS-CoV-2. And this example is how one nucleotide change in this antiviral gene is really important for protection of West Nile. So, as I told you, these viruses hide in the immune system, right? Within a cell, hide from the immune system. And I gave you an example of alphaviruses. Here I will talk about picorna and flaviviruses. Because of the time, I will just restrict my data to West Nile so that you can see how these viruses trick the immune system and hide in our cell, and how our host is evolving to find out where they are, okay? So the example is a gene called OAS1. It's called oligoadenylate synthetase. And this gene, what it does is the protein detects the viral RNA, and then makes a second messenger called 2,5A. And this 2,5A brings in a latent RNA cell, and they dimerize and go and start cleaving the virus and the host RNA. The idea is to kill the cell. If the cell is infected, this gets activated and kills the cell, so that the infection does not spread in the body. So it's a very strong innate immune response. But when we got into this, we found something odd. What we found was there was this one nucleotide change just before the 6th exon, and important for splicing. And this SNP was really important for clearance of West Nile virus. And that was all what we knew. And what was known as this one nucleotide change can make 2 different splice forms. Okay, there are multiple splice forms, but these are the 2 major splice forms. If you have an A, what you get is P42. And in the P42, you have 5 exons, a little bit more, but not the 6th exon. But if you make P46, you have the 6th exon. And it's called P42 and P46 based on the molecular weight of the protein. Okay, so when you have OAS P42 made, what you see is, if you infect a cell with West Nile virus and you overexpress P42, you don't see a difference in the viral control. You can't control the virus. But if you have P46, it suddenly has a log protection. That shows that P46, just one nucleotide, can have such an enormous effect on clearance of the virus. So we wanted to ask why. And we had learned a few things from ZAP. And readily what we found, which I'm not showing you here, is P46 has a CAX box. And I told you, if you have a CAX box, it goes to the membrane. But this time, P46 did not go to endolysosome. Now P46 went to the Golgi membrane. So this is stained with the Golgi, and you can see it is exactly in the Golgi, right? And P42 is all over the cell. It's not, but it's not in the Golgi, right? So what we found is the location of these 2 proteins. These are 2 isoforms. One nucleotide change caused these formation of these 2 isoforms, and they're going to 2 different regions, right? So then we asked, okay, we know P42 does not have a CAX motif, the CAX box, right? So if we add a CAX box into P42, what will happen? Will it change its location? And it does. All the OS1 which is around the cell now gets into the Golgi, and it is concentrated in the Golgi, right? And we went and mutated this CAX box, and now the Golgi localization is lost in P46. It's all over the cell, right? So this was intriguing. Now we asked, is this any way, shape, or form important for West Nile? Because we know P46 controls West Nile really well compared to P42. Okay, so P42 is all over the cell, and you see West Nile uses ER and Golgi membranes, and you see that West Nile is in these pockets, right? I showed you they kind of are in a hive-like structure, but P46 is all over the cell, right? But in the case of P46 is in the Golgi. That's exactly where the West Nile are forming these viral replication organelles. And you can see here the colors here. You see it is superimposing 3 different colors, and you can see that West Nile is exactly exactly in the replication complex or around the replication complexes of West Nile, right? And that's how they find the viral RNA and activate RNase-L and chew it up, right? Now the question was, I told you this, the SNP which we are talking about, if you have a G, you'll get P46. If you have A, you get P42. So we asked, is this important in West Nile or Zika infection, which are the major flaviviral infections which threaten the world? And what you see is just very intriguing. The P46, G producing P46 is overrepresented in Africa. The A, which is susceptibility for P42, that means to say P42 doesn't help us, is overrepresented in Americas, Europe, Eastern Asia and South Asia too, right? That means to say, African population are really resistant to Zika and West Nile, but the rest of the world is not. And we see this exact thing around the world. Zika and West Nile are spreading in the Americas, spreading in Europe, in East Asia and South Asia, but not in Africa. In Africa, West Nile virus is not a problem, or Zika is not a problem. Yes, Ebola is a problem, but not these two viruses. So this shows how there are single nucleotide polymorphisms which have tremendous effect on how we control these viruses. So if you got lost in some of these data, what I want you to remember is these antiviral proteins, which are induced by interferons, are located in different membranes. Some are in the Golgi, some are in the ER, some are probably on the plasma membrane, some are on the nuclear membrane, or in different locations. And they're waiting for the viruses to get in so that they can find them and neutralize them, right? And our DNA is constantly modifying, right? So that we can fight these viruses by making alternative splice variants, which are functional, which have better activity against these viruses. So the take-home message, our DNA is constantly evolving. When I say constantly evolving, it is not like the viral RNA or viral DNA or bacterial RNA or DNA, because because they have a very high replication rate, like 20 minutes, right? So they have much more faster pace in evolution, but not humans. It takes generations for us to evolve resistance or susceptibility. So identifying these specific changes can help us come with new therapies. So I will stop there. Maybe I went 5 minutes over. Thank you very much, Shubha, and everyone else. And I'm happy to take any questions.
Thank you, Ramsavan, for a very exciting and very interesting talk. And there are a couple of questions in the chat box. Maybe I will read out, or can you see them?
Let me check. Yeah, I think I can see them.
Okay, could you please answer those two questions?
Okay.
He's seeing. Shall I read out?
Yeah, the first question is, what is the state of longevity of interferons type 1 and type 3 in an infected cell? What elemental status govern them? This is a great question. So type 1 interferons are made very early. The minute your cell sees infection, viral infection, in 3 hours or 2 to 3 hours, you see type 1 interferons being made. And they also signal back and signal onto other cells, the neighboring cells, to alert them that there is virus infection around. And most of these responses are cleared in 12 to 24 hours because you don't want interferons to be around for long. It causes autoimmunity. Lupus is an interferon-mediated disease, so you don't want these interferons to be around. But on the other hand, type 3 interferons are slightly delayed. They start somewhere around 12 to 24 hours. And their expression as well as induction ends around 72 hours. So if you consider the timeline, it is very early. The first few days when you feel sick, that's because of interferons. And once the interferon wanes off, you stop feeling sick. Okay. Do the hidden viruses have any possibility of being mutated by any means? Oh yeah. So the best example is hepatitis C virus. So hepatitis C virus, when it infects us or infects a person, it has one genome, but it starts constantly mutating. And good example is flu. The other example is HIV. That is why for these, it is hard to make vaccines, right? They keep on mutating. Okay, some TLRs are localized in organelle membranes. How these TLRs fail to recognize RNA of alphavirus? Great question. It depends on which endosomal membrane it is. Yes, I'm sure alphavirus is detected in these endosomal membranes, right? And induces interferons, right? So, but for example, in ZAP is already there along with the TLR, presumably. We have not tested it, right? But TLR induces interferons, and then interferon comes back or alerts the neighboring cells. But ZAP is already present in the cell before interferons are seen. That means to say they're the primary restriction factor, especially for alphaviruses. Can coronavirus infection be, oh, there's another question. Are type 1 interferon expression and cytokine storm related? Yes and no. Type 1 interferons can be in this case, for coronavirus, the cytokine storm which we're thinking about are the cytokines which are induced in macrophages and monocytes like IL-1, IL-6, TNF. They have not seen type 1 interferon in the cytokine storm. If there was type 1 interferon induced early on, it would be actually really good to curtail the virus. I think those are the questions I see. Can coronavirus infection be endemic? I don't know what you mean there, but one of the coronaviruses have been there for a long time, like with kids, right? Most of the kids have some sort of coronaviruses around, but they have been very benign, right? But the coronavirus, what we see now is highly pathogenic. It's SARS-CoV-2. That was not endemic in the human population, if that is the question Prakash is asking. Those are the questions, and I think there are no more questions there.
Okay. Thanks a lot, Savan, for being with us, and it was a very good interactive session. Now I request our chairperson to thank you.
Hello, Dr. Savan. I thank you very much for your highly motivating talk on behalf of the University of Mysore, on behalf of the microbiology department, and also on behalf of the AMI, that is Association of Microbiologists of India, Mysore chapter, and all our audience. On behalf of all our audience, I thank you very much.
Thank you so much. I appreciate it.
Yeah, thank you, Savan. Bye.
Nice.
My dear students, please stay tuned until with us at 11:30.
Next to 11:30.
11:30. Hatumukal. Maybe.
Maybe another 45 minutes.
Yes, they should. Our work.
ಇದಕ್ಕೆ ರಾಘವನ್ ಹಾಗೆ ಇರಲಿ